Thank you, Ashley. For today's webinar, I want to begin with just a brief overview of what the In-Cell Western Assay is. From there, I want to spend the majority of today's webinar going over specific examples of how research groups like your own have used the In-Cell Western Assay in their own research. This will include examples in protein phosphorylation, apoptosis, RNAi analysis, and cell surface protein analysis. The In-Cell Western Assay begins by seeding your cells of interest into a microtiter plate. These plates can range from 6-well to 1536-well plates. The only requirement is that they have clear well bottoms. Once your cells are seeded and they've grown to the desired confluency, you perform your treatment. As we'll see over the course of the webinar, the type of treatment you may use will vary depending on your application for the In-Cell Western Assay. Now, once your treatment is finished and you remove the media, you then fix and permeabilize your cells. Once permeabilized, you then add your primary antibody against your target of interest, and then you add secondary antibodies, which are conjugated to IRDyes. IRDyes are fluorophores that fluoresce in the near-infrared wavelengths, and this ensures you have low background and high sensitivity for these assays. Once you've added your secondary antibodies and you've washed your plates, you then go ahead and scan the plates on an Odyssey system. Compatible Odyssey systems include the Odyssey Classic, the Odyssey CLX as shown, and the Odyssey SA as shown. Now, the In-Cell Western Assay offers a number of benefits when performing these types of experiments when compared to traditional Western blotting. The first of these benefits includes the protocol itself. When you're preparing these types of experiments with traditional Western blotting, you're required to do cell lysate preparation, running of SDS-page gels, and transferring those gels and protein of interest to a membrane. And then finally, you begin with blocking and antibody incubation steps. All of these steps can be very time-consuming for a researcher. Now, for the In-Cell Western Assay, we eliminate all these steps so we can save you time in the protocol. Additionally, the In-Cell Western Assay saves you time because you can generate data in a high-throughput manner. If you were to perform an In-Cell Western Assay with a 384-well plate, that is 384 data points you can obtain in a single day. To generate that many data points with traditional Western blotting, you need to run roughly 43 10-well gels. Now finally, the In-Cell Western Assay also benefits over traditional Western blotting by increasing the quality of that generated data. To highlight this, here's some data from Aguilar et al. Published in 2010 from PLOS One. What they did is they tested and compared the reproducibility from both Western blotting and the In-Cell Western Assay. Shown here are the results. For both assays, they analyzed the same sample in multiple lanes and gels for Western blotting, or in multiple wells for the In-Cell Western Assay. They performed each assay and then quantified each replicate, and compared the quantitative value from each replicate to the relative mean of the dataset. Shown here are the results. In the top two graphs, they were probing for GAPDH, and the bottom two graphs, they were probing for PMLC20. For both targets, if we look at the data, you can see the reproducibility for all the replicates with the In-Cell Western Assay hovers very tightly around that relative mean. What this means is you have very high reproducibility from replicate to replicate, experiment to experiment. When we compare this to traditional Western blotting, we can see that each of the replicates with traditional Western blotting can have very high variance around that mean for the dataset, showing you have low reproducibility for Western blotting, high reproducibility for the In-Cell Western Assay. So in addition to improving this quality of this generated dataset, you increase the amount of data you produce and you save time in that protocol. Now from here, I want to spend the remaining part of this webinar focusing in on specific examples of how research groups have utilized the In-Cell Western Assay in their own work. So first, let's go through some specific examples looking at protein phosphorylation. Now, if you want to use the In-Cell Western Assay to study protein phosphorylation, there are a couple things to take into account. The first is, what's your target of interest? Well, for the protein phosphorylation application, your target of interest will be phosphorylation. So you want to ensure you have a primary antibody directed against that phosphorylation site. It's important to ensure that that primary antibody is specific, because if it's not, that will affect your sensitivity for the assay. To detect your primary antibody at light core, we would recommend to use IRDye 800CW conjugated secondary antibodies. IRDye 800CW is optimized at light core to work with the In-Cell Western Assay. and it's designed to work with the 800 channel on an Odyssey system. Now, for these assays, it's important to normalize your phosphorylation signal from well to well to take into account any changes in cell number. To accomplish this, there are a number of techniques to choose from. One option is you can use primary antibodies directed against your total protein, or perhaps a housekeeping protein like actin. Now, for these methods, you'll want to use IRDye 680RD conjugated secondary antibodies. IRDye 680RD is optimized at LigCore to work with the In-Cell Western Assay and is designed to work with the 700 channel on your Odyssey system. Now, if you wish to forego the need to use those primary and secondary antibodies for normalization, you can go ahead and use CellTag 700 stain. CellTag 700 stain was developed at LigCore and allows you to non-specifically stain your cells and allows for easy normalization from well to well for the In-Cell Western Assay. Now let's go through a specific example of a group using the In-Cell Western Assay to study protein phosphorylation. This data was published by Pierre Eugene et al. In 2012 from PLOS One. Now, this research group focused their work on both the insulin receptor and the IGF-1 receptor. In cells, the insulin receptor forms homodimers with itself. The IGF-1 receptor also forms homodimers, and insulin receptor and IGF-1 receptors also form hybrid receptors with one another. Now, if you look at insulin, insulin has high affinity just for the insulin receptor homodimers, whereas IGF-1 has high affinity for IGF-1 receptor homodimers, as well as the hybrid dimers. Now, for a diabetic patient, they may not be injecting insulin to regulate their glucose levels, but rather an insulin homolog. These homologs have been studied extensively for their actions at the insulin receptor homodimers, as well as the IGF receptor homodimers, but before this work, their action at the hybrid receptors was completely unknown. Now, these hybrid receptors have implications in a number of disease states, so any nonspecific interaction between these therapeutics and these hybrid receptors could have very large implications for a patient. So, to study this group examined insulin, IGF-1, and a number of insulin homologs. In initial screening, they identified the insulin homolog glargine as having abnormal affinity to these hybrid receptors as compared to insulin. So what they did is they used glargine, as well as two of its metabolites, M1 and M2, in downstream studies. They wanted to study these two metabolites, as glargine has been shown to be quickly metabolized to M1 and M2 after injection. So they first examined the affinity of IGF-1, insulin, glargine, and the two metabolites M1 and M2 in a Brett binding assay. This assay revealed, as we can see in these graphs, that the affinity of glargine is much higher for these hybrid receptors than insulin. But what was interesting is when we look at these hybrid receptors, the two metabolites M1 and M2 have lower affinity for the receptors when compared to glargine, and that their affinity is much more similar to insulin itself. From here, they wanted to examine a number of downstream effectors. This included looking at phosphorylation of AKT and ERK. So to monitor phosphorylation of these targets, they went ahead and they utilized the In-Cell Western Assay. So what they did is they incubated their cells of interest, expressing their hybrid receptors with different ligands and different concentration of these ligands and replicates. By utilizing the In-Cell Western Assay, they could quickly and easily monitor phosphorylation of either AKT or ERK with phosphospecific primary antibodies. And when they applied either insulin, IGF-1, glargine, M1, or M2 to these cells, they could read an increase in phosphorylation of both AKT and ERK. And so shown here are two example Western blots from these experiments, as well as the data generated by the In-Cell Western Assay. Now what they found is that glargine had a higher level of potency for both ERK and AKT activation towards these hybrid receptors. But what was interesting is that its two metabolites, M1 and M2, had potency levels that mirrored insulin. So this showed that although glargine had higher affinity for the receptors and higher levels of activation for these hybrid receptors, its active metabolites in vivo matched the action of insulin more closely. So for this group, they're able to use the In-Cell Western Assay to monitor protein phosphorylation of both AKT and ERK. And by utilizing a plate-based assay, they could easily vary the concentration of a number of ligands to quickly determine what are the EC50s for these compounds for activation of these downstream pathways. So let's move on and look at Another example of an In-Cell Western application focusing with apoptosis. Now, when you set up the In-Cell Western assay to measure apoptosis, there's a couple things you're going to want to consider. The first is what exactly your target of interest is going to be in the assay. As we'll see in a few moments, the target of interest read in the In-Cell Western assay can vary depending on your system. So it's important to choose one that's validated and linked with cell viability and apoptosis in your system. Some companies like Mitosciences offer detection kits for caspase-3 and PARP1. Now these are early apoptotic markers and can be read in the In-Cell Western assay and can be used to measure apoptosis. Now, regardless of what your final target is, at Lycor we would recommend to use IRDye 800 CW conjugated secondary antibodies for detection of that target. So let's go through a couple examples in research, and the first example will be some data published from Possimo et al. From 2013 in PLOS One. Now, this research group focused on developing a neuronal viability assay to study neurodegenerative diseases. Specifically, they wanted to see how protein toxicity affected cortex cells, and they wanted to look at allocortex and neocortex cells. So they utilized two side-by-side assays to accomplish this. The first is a plate reader assay to measure ATP levels. The second is an In-Cell Western assay to measure microtubule-associated protein 2, or MAP2 levels. Now, it's been shown by Mullet et al. In 2007 that performing an In-Cell Western assay and monitoring MAP2 levels can accurately measure neuronal viability. So to start, they wanted to go ahead and correlate cell numbers to the signal they could read for each of these assays. So what they did is they seeded their cells between 40,000 and 120,000 cells per well. Then they performed either the MAP2 In-Cell Western assay, which is shown on the left-hand side, or the ATP assay shown on the right. What they found is that the In-Cell Western assay provided accurate readings regardless of the cell density used. In contrast to this, the ATP assay only worked well at lower cell densities and struggled as you got to higher cell densities. Now from here they wanted to test how these cells would react to oxidative stress. So to accomplish this, they seeded either 100,000 cells of allocortex or neocortex cells, and then incubated those cells with increasing concentrations of hydrogen peroxide. They then performed either the MAP2 In-Cell Western assay or their ATP assay. Both assays were able to find that neocortex cells were more susceptible to oxidative stress than allocortex cells. Now this was surprising to this group because if you traditionally look in vivo, the allocortex cells are typically more susceptible to protein toxicity than these neocortex cells. So to test if the allocortex cells were indeed more sensitive to protein toxicity in these assays, they performed a proteotoxic stress test. For this, they added either MG132 or PSI, and these are inhibitors to the proteasome, and their application mimics a protein toxicity state. With both assays, they found that allocortex cells were more susceptible to proteotoxic stress than neocortex cells, which is consistent to what's seen in vivo. They confirmed the result of their MAP2 In-Cell Western assay with immunofluorescence shown on the right-hand side. And the immunofluorescent results show indeed that allocortex cells are more susceptible to protein toxicity than neocortex cells. So for this group, they were able to use the In-Cell Western assay to measure MAP2 levels, which correlates to neuronal viability. And they even found that performing the In-Cell Western assay provided accurate results regardless of the cell density they used. And traditional methods using a plate reader failed at higher cell densities. Now let's work through another example using the In-Cell Western assay to measure apoptosis. And this will be from Jamin et al., published in 2013 from PLOS One. Now, this group wanted to study the role of diet in the formation of colon cancer. And specifically, they looked at BAP, which is a polycyclic aromatic hydrocarbon, and PhIP, which is a heterocyclic amine. Now, both of these are present in cooked meat, and they wanted to see how these different compounds affect genetic mutation, specifically at the APC gene. Now, this APC gene has been linked to early formation of colon cancer when mutated. Now, in this paper, they performed upstream studies where they applied these different compounds and used mass spectrometry to determine that these different compounds form DNA adducts at the APC gene in a dose-dependent manner. So they then went on to study these compounds a little bit further, and this is where they used the In-Cell Western assay. and they looked at two different cell lines. They looked at a cell line sensitive to mutations at the APC gene, which is denoted as APC-min, as well as wild-type cells. And then they utilized the In-Cell Western Assay to measure two things. The first was phosphorylation of histone H2AX. Now, it's been shown previously that increases in phosphorylation of this histone are directly linked to global genotoxicity inside the cell. Now, they also used the In-Cell Western Assay to measure DNA amount or DNA content of these cells, which is directly linked to viability of the cells. So they first wanted to look at the effects of BAP on these different cells, and shown here are the results from their In-Cell Western Assay. Now, when we look at this graph, on the x-axis is the applied concentration of BAP. On the left side for the y-axis, we have the quantified amount of phosphorylation for histone H2AX as measured by the In-Cell Western Assay, and this axis will correlate to the bar chart. On the right y-axis is our cell viability as measured by our In-Cell Western Assay DNA stain, and this axis will correlate to the traces shown. For BAP application, they could readily detect a dose-dependent increase in histone phosphorylation and an increase in cell death. And they found that the APC-min cells were more vulnerable than wild-type cells. So this suggested that indeed, BAP mutations at this gene result in both genotoxicity and cell death. From here, they then wanted to look at PhIP. And when they applied PhIP, they found similar results. They saw a dose-dependent increase in both histone phosphorylation and an increase in cell death as read by the In-Cell Western Assay. Finally, they wanted to examine both compounds together with the In-Cell Western Assay. So for this, they looked at either PhIP alone, BAP alone, or both together at a 10-to-1 ratio. Now, they chose this ratio because this is the ratio you'll typically see in cooked meat, so they found that both compounds can increase histone phosphorylation. And we can see this here as read by the In-Cell Western Assay quantifying phosphorylation of our histone. But what was interesting is that both compounds together did not have a significant difference in histone phosphorylation when compared to BAP alone. So for this group, they were able to utilize the In-Cell Western Assay to monitor two events linked to apoptosis. The first is phosphorylation of histone H2AX, which is linked to genotoxicity. The second is DNA content, which is linked to cell viability. Now let's go through an example in research looking at RNAi analysis. If you want to utilize the In-Cell Western Assay to measure and look at RNAi, your first consideration will be your readout. Do you want to treat cells with RNAi and monitor how knockdown of a certain gene affects phosphorylation of a target protein? Or do you want to see how it affects expression of a target protein? Regardless of your readout, at Lycor we would recommend to use IRDye 800CW conjugated secondary antibodies. For an RNAi screen, it's important to normalize your signal from well to well. To accomplish this, you can use a primary antibody directed against a housekeeping protein such as actin. Now with this approach, we would recommend to use IRDye 680RD secondary antibodies for detection of that primary. If you wish to forego that need to use a primary and secondary antibody, you can easily go ahead and normalize from well to well based off of cell number using cell tag 700 stain. Now let's see how a group used the In-Cell Western Assay for an RNAi screen. This data is published from Bond and Foley in 2009 from PLOS Pathogens. Now, this screen focused on the IMD pathway, which has high homology to the TNF pathway in mammals. And a hallmark of activation of this pathway is junk phosphorylation. Now, this junk phosphorylation is tightly regulated, and the mechanism by which it's regulated is unknown. So to identify modulators of junk phosphorylation, this group set up an RNAi screen utilizing the In-Cell Western Assay. So what they did is they incubated cells with over 15,000 double-stranded RNA molecules, and they did this one at a time. Now after incubation for 3 days with their double-stranded RNA, they then added a ligand, PGN. This ligand will activate the IMD pathway and result in junk phosphorylation. From here, they removed the media, fixed, permealized their cells, and probed for phosphojunk in the 800 channel on an Odyssey system, and actin in the 700 channel on an Odyssey system utilizing the In-Cell Western Assay. Performing this screen, they knew they would identify 3 types of genes. First, knocking down an essential gene would cause cell death, and this would result in both a loss of signal of actin staining and phosphojunk staining. Second would be knockdown of a suppressor of junk phosphorylation. And what you would see in the assay is increased levels of phospho-junk signal with no effect on actin signal. And the final class would be a normal enhancer of junk phosphorylation, where you would see in the assay a decreased junk phosphorylation signal with no effect on actin. So when they performed the assay, they could readily identify these three genes. And shown here is an example of an In-Cell Western Assay image. Shown on the left is their phospho-junk signal, the middle their actin, and on the right is the merge of the two. And here they could identify CLK and ACT79B as essential genes. And they could also identify a known enhancer and suppressor to the IMD pathway, DREAD and CKA respectively. So from here, they analyzed the full results of their RNAi screen, and what they did is they ignored their essential genes and only looked at the enhancers and suppressors. And they performed the screen twice, once with just 15 minutes of ligand activation and a second with 60 minutes of ligand activation. And the results are shown here in a graph. And in these graphs, what they do is they align the double-stranded RNA from strong suppressors on the left to strong enhancers on the right. Now for both of these two graphs, the y-axis is a z-score. The positive numbers you see reflect increases in junk phosphorylation, and the negative numbers reflect decreases in junk phosphorylation. The red dotted lines you see signify significance for these assays. From this screen, they could identify a number of junk modulators. From here, they wanted to confirm these results with additional In-Cell Western assays. In this first graph we see here, what we're examining are a number of potential enhancers and suppressors to junk phosphorylation. Included are some negative controls, CG11318 and TOL, and these have no effect on junk phosphorylation. We also have two positive controls, DTAC1 and Key, which are a known enhancer and suppressor to junk phosphorylation. What we can see for each of these potential modulators, followed with knockdown of these target genes and activation with ligand, is we could readily confirm our RNAi results. Now, one question was whether these results we see here are due to changes in junk phosphorylation, or if they were actually changes to total junk expression, which could explain why we see changes in phosphorylation levels. So to test this, they repeated the In-Cell Western assay, but rather than normalizing their phospho-junk signal to actin, they went ahead and normalized it to junk. When they did this, they could readily confirm that all of these potential enhancers and suppressors were indeed junk phosphorylation modulators. Now from here, they chose one target, PVR, and examined its role in junk modulation further. So what they did is they went ahead and reutilized their RNAi screen, and they looked at the entire PVR pathway for its effects on junk phosphorylation. Now the PVR pathway is normally activated by the ligands PVF1, 2, and 3, and upon activation results in a downstream signaling pathway including GRB2, SHP2, SOS, RAS, RAF, MEK, and ERK. Now shown here is this pathway, and it's colored with the results from the RNAi screen. Now for the coloring, a strong red color is indicative of junk suppressors. And what we can see is that the entire PVR pathway appears to be a suppressor of junk phosphorylation. Now as a control, they also monitored an unrelated pathway to junk, which is the wingless pathway. And when they analyzed the RNAi screen results for this wingless pathway, we could see that there's no strong association, positive or negative, with junk phosphorylation. So from here, the group was able to further confirm the results from the In-Cell Western assay by using techniques like qRT-PCR, Western blotting, and other pharmacological tools. So for this research group, they were able to utilize the In-Cell Western assay to directly monitor phosphorylation of junk in an RNAi screen and quickly identify novel regulators to their pathway of interest. So why don't we work through our last example in research, which will be focused on cell surface protein analysis. But before we go through these examples, why don't we pause really quickly just to take a quick poll. Ashley?
Thanks, John. I've launched the poll, so our audience should see the question. And the question is, the on-cell Western differs from the in-cell Western in that you use suspension cells, you don't fix cells, or you don't permeabilize cells. We'll give everyone just a couple of moments to choose an answer and hit submit, and then we'll move on. Looks like we have about 25% of our audience having participated right now, so we'll try to get that up to maybe about 75% before we close the poll out here. All right, we're getting really close to 50%. Give you guys just a couple more seconds to submit your answer. And you should be able to do that right on your screen. Okay, why don't we close the poll and let Johnny give you the answer.
Great. It seems like a good number of you are familiar with what the In-Cell Western Assay is. The correct answer here is B. The In-Cell Western Assay differs from the In-Cell Western Assay in that we do not permealize our cells. Now, if we don't permealize our cells for these assays, then our target of interest we're probing for needs to be extracellular to the cell. Now, the In-Cell Western Assay can be used to study a number of different targets. The first is we can look for a ligand of interest binding to a receptor. In order to do this, what we need to do is go ahead and label our ligand with an IRDye, and then we can utilize that labeled ligand to test whether or not it's binding to our receptor of interest and go ahead and calculate things like affinity. Now, one key question here is, how do I label my ligand of interest? And there's a number of ways to do this. At Ligcore, we offer a number of our IRDyes with reactive chemistry groups. And what this will allow is for you yourselves to label your ligands in the lab with an IRDye. If you want information on these dyes, please visit ligcore.com/IRDye. Now, as an alternative, Ligcore offers a custom services program. Now, here you can talk directly to a Ligcore scientist and determine the feasibility of a labeling project. And then from there, either you can go ahead and label this yourself, or Ligcore can actually label your ligand for you. And if this sounds interesting to you, please visit ligcore.com/custom services for additional information. Now, in addition to ligand binding, you can also study protein localization to the plasma membrane with the In-Cell Western Assay. For this, you want to ensure that you have a primary antibody that binds to a motif that is extracellular to the cell. Once you have such a primary antibody, you'll be able to use it to bind to that motif, and it will only be able to bind to that motif when your protein of interest is localized to the plasma membrane. Now, if your protein of interest is internalized, that motif will no longer be accessible to your primary antibody with the In-Cell Western Assay, and what you'll detect is a loss of signal with the In-Cell Western Assay. Now, as with the In-Cell Western Assay, you want to make sure to normalize your signal from well to well for these plates. Now, since we're not permealizing our cells, that can get a little tricky. So, to normalize, you have a couple options. The first is to use cell tag 700 stain to normalize to cell number. Now, please note, cell tag 700 stain is a non-permeable stain. So in order to use the stain, what you need to do is first incubate your cells with primary and secondary antibodies. And once you're done with that secondary step, you then permealize your cells and then incubate with cell tag 700 stain. An alternative to using cell tag 700 stain is using IRDye 680RD NHS ester. This IRDye is a reactive IRDye and will non-specifically label the proteins on the cell surface in the In-Cell Western. And this will allow you to normalize from well to well based off of cell number. Now let's walk through some examples in research. And the first one will be from Milovankev et al., published in 2013 from BMC Veterinary Research. Now this group wanted to look at how the cell surface proteomes differed between normal and cancerous cell lines. And specifically the cancer cell line they wanted to look at was an osteosarcoma cell line, or OSA. So to accomplish this study, what they did is they took whole cells, either wild type or OSA cancer cell lines, and biotinylated cell surface proteins. Once biotinylated, they then immunoprecipitated those biotinylated proteins and analyzed the immunoprecipitate with mass spectrometry. From this screen, they could quickly identify a number of protein hits that differed in their cell surface expression compared to wild type, or the CNOB cells, to cancerous cell lines, the POS or HMPOS cell lines. Here, they're able to use qRT-PCR and Western blotting to confirm that the expression level of these proteins differed between these different cell lines. But what these experiments do not test is whether the plasma membrane localization changes between the cell lines. So to test for this, they needed to utilize the In-Cell Western Assay. For the In-Cell Western Assay, they seeded their cells of interest and then incubated whole live cells with a primary antibody directed against their protein of interest. They then incubated those cells with a secondary antibody to visualize their target on an Odyssey system. From here, they then stained their cells with Hoche, allowing for normalization to DNA from well to well. Here, they're able to perform the In-Cell Western Assay looking at CD44 between wild type and cancer cell lines. And they could readily detect differences between the three cell lines. What we can see here is that the cancer cell lines, POS and HMPOS have higher amounts of that CD44 localized to the plasma membrane when compared to wild-type cell lines. So for this group, they were able to utilize the In-Cell Western Assay to monitor protein localization to the plasma membrane. And this allowed them to monitor how localization changes depending on the cell line you're looking at. So let's walk through our last example of the day, which will focus on ligand binding. And this work is from Sexton et al., published in 2009 from PLOS One. Now, this research group wanted to develop a molecular imaging agent for use in vivo. And they wanted to utilize the near-infrared wavelengths to accomplish this. And so the question is, why near-infrared? Well, when you're working in the near-infrared wavelengths, like those you see on an Odyssey system, the absorption of light from hemoglobin and water is greatly reduced. And here in this graph, we can clearly see this. And so in this graph, we're looking at different spectrums of light. And at visible wavelengths, those below 650 nanometers, we can see two traces which represent hemoglobin's absorption of light. And what we can see is at these visible wavelengths, hemoglobin absorbs the majority of our light intensity. And this will decrease our sensitivity for these types of assays. Now, if we go beyond the near-infrared wavelengths, beyond 850 nanometers, we can start to see that water becomes a problem and also starts to absorb light intensity. Now, this fixes the Odyssey system and the Odyssey wavelengths in a sweet spot, where we've minimized the effects of both hemoglobin and water, yielding optimal sensitivity for these types of studies. Furthermore, IRDye 800CW is a non-toxic dye, and working with this dye allows you to have very translatable work to a clinic. So in this example, they wanted to develop a molecular imaging agent against TSPO, or the mitochondrial translocator protein. Now, it's been shown that this protein is involved in a number of disease states, and a sensitive IRDye 800CW ligand could prove very valuable. To develop this ligand, they went ahead and took two TSPO ligands and labeled them with IRDyes. They then validated the ligand affinity with radioligand binding studies, and then examined how these ligands bound to whole cells. Here we can see the results from their radioligand binding studies. They constructed two IRDye 800CW ligands, NIR-ConPK and NIR-6T. From these radioligand binding results, they found that the addition of IRDye 800CW affected the affinity of NIR-ConPK for TSPO, whereas it appeared not to affect the binding affinity for NIR-6T. From here, they then want to see how effective these compounds were for looking at their target of interest in cells. And they utilized the In-Cell Western Assay for this. So what they did is they took both their low affinity NIR-ConPK ligand as well as their high affinity NIR-6T ligand, and they first incubated each of these ligands to whole cells and examined ligand binding after certain time points, ranging from 10 minutes up to 4 hours. And the results are shown here. And what they found is that their low affinity ligand quickly hit a steady state of binding after about an hour of incubation. Their high affinity ligand, though, never reached that steady state. Next, they wanted to look at how did incubation of these cells with a specific unlabeled TSPO ligand affect binding of their IRDye 800CW ligand. So what they did is they performed their In-Cell Western Assay, and with these different ligands and their unlabeled compounds, and they found that the low affinity ligand was easily competed off with the two TSPO ligands, whereas that high affinity ligand could not be competed off. So from here, this group was able to utilize this data and hypothesize that although the NIR-ConPK might have low affinity to TSPO directly, it appears to be identifying a novel, unidentified TSPO homolog in whole cells. On contrast, their NIR-6T ligand, which had high affinity in vitro binding assays, appears not to be a very useful tool for in vivo studies. So for this group, they were able to utilize the In-Cell Western Assay to monitor and validate ligands of interest labeled with near-infrared dyes like IRDye 800CW. Now from here, if you went ahead and labeled your own ligands and did these validations, you could easily then move into your in vivo studies to study a biology or a system of interest. Now that concludes our examples for this webinar, and to give a brief summary, the In-Cell Western Assay provides a flexible platform for researchers to perform a number of applications, and to generate data in a high quality and high throughput manner. Now for additional information on the assay, please make sure to visit Lycor.com/ICW. Now at this point, why don't we take a couple questions from the audience? Ashley?
Sure, John. The first question we have is, what is the best way to find a specific antibody?
That's a great question. Primary antibodies on the market may not work well for these types of assays. Now, when you're choosing a primary antibody, make sure that the primary antibody is validated for immunohistochemistry work. Now, once you have that validated primary, you want to make sure that it's specific using a Western blot. Now, if you have a primary antibody that has non-specific bands, you want to optimize that primary antibody by optimizing things like your blocking conditions. Now, once you have that primary antibody that's been validated for immunohistochemistry and is specific towards your target, then you can go ahead and move into the plate-based assay. I believe we have time for just one more question. Ashley?
Yeah, the last question that I have here is, how can I tell if your IRDyes can label my ligand of interest?
Another great question. If you aren't sure you can label a ligand with an IRDye, you should contact Licor's Custom Services at licor.com/custom services. Once you visit this website, you can input your information and talk directly with an IRDye chemist, and they'll be able to let you gain more information of how to label, the feasibility, and sort of the methods of how to go about labeling your ligand of interest. Well, that's all the time we have for today. Thanks again for attending the webinar, and good luck with your In-Cell and On-Cell Western assays.